My name is Dr. Brenna Hill, and we're in the Vaccine Research Center in the Human Immunology section of the building. I look at immune responses after vaccinations against HIV. My great-great-grandfather back in Ireland taught science, so it's kind of been in the family, but I'm the first one in the family to actually get into science. When I was in roughly third grade, we used to have newsreels, in 1983 they were announcing a new disease that was affecting a population in San Francisco.
At first it seemed to strike only one segment of the population. Now, Barry Peterson tells us this is no longer the case.
There is a 1 in 5 chance a victim will die within the first year of the illness, a disease experts are now calling a national epidemic.
And it was really interesting because it was a viral disease where the virus knew to attack the immune system to keep itself alive, and it just absolutely fascinated me. And ever since then I just kind of got into doing science and biology and anything that was going on for treatments. I emailed a particular lab that was doing HIV research. That was really fascinating. I emailed, said I was interested in doing that, and they took me on, and I've been with them for the past 15 years. We are working towards a goal. We're working towards getting a vaccine to HIV, and HIV is a very complex virus. It likes to mutate itself to evade the immune system as much as possible, but the biggest thing is to determine if the vaccines that we have out there now are going to be even helpful. It is baby steps. And so I usually get samples from patients who either have the disease or they were a volunteer in our vaccine trials, and we look at the immune response that happens during the disease or after the vaccine response and decide, is it the kind of immune response that could be helpful? So I do a lot of work in what's known as flow cytometry, where we take immune cells and we label them with fluorescent antibodies. And then we look at what each one of them does. So you can see this spray of cells right here, which is showing that those cells are actually responding to the antigens of the vaccine, and it's showing that these cells are actually doing some good work. So we can look at both blood, lymph nodes, and every different kind of tissues in the human body. You've got a 96 well plate, and your samples are in there stained and ready to go. Then after it picks up the sample, it injects it into this device, which then moves it into the larger device. Inside are lasers that are shooting at each individual cell and looking at the fluorescence of each individual cell. And that is being translated and then recorded by the software. Everybody here, at least in this lab and in this building, we work with each other.
So what you guys do this weekend?
We're constantly talking to each other, sharing ideas, and if we don't have the expertise, we will reach out to anybody else at the NIH. We work so closely together, and we do a lot of hard work. The technology that we're developing is helping us get faster and faster coming to a vaccine goal. I would say we're about halfway there, and hopefully in the next 15 years we have something that's even better. Just don't give up. If this is something that you love, just keep going with it. I love it every day. I've been doing it for a good 17 years, and I still love coming in every day, doing stuff at the bench, getting the results and seeing what happens.